All right, thanks for coming along, everyone. It's the 9th of August, 2022, and this is the Open Source Antibiotics Mur Ligase meeting. And we have a few people here who are not on vacation, which is awesome. I did generate an issue just now for this meeting, which I will now share. Just in case people don't have it, and then we can start receiving updates from people as and when. Okay, here we go. So that's a bit small. Okay, here you go. Just to update people, so I went about a week ago to the Gordon Research Conference on new antibacterials in Italy, and it was a really good meeting, very interesting. Lots of people doing some really fascinating things, and there was some policy people there from GARD-P and all these places. So it was a good, lots of science, which was really great, and people were certainly interested in what we were doing, our model and what we were doing. I'm following up on a couple of specific things. The two big things that came up really, one was data that Chris Dowson and LifeArc had generated. Not just the previous project that Chris and LifeArc have been involved in, which I think is what we've been discussing, but some fragment screening campaigns that LifeArc had been doing versus conserved bacterial targets. And they'd done a huge amount of work, which I wasn't aware of, expressing proteins and getting fragment screens to work, and had prioritized sort of 50 targets over the last few years. And all of our favorite Mur enzymes are in there. So they've done screens versus all this, yes, and are happy to share all the data. So I guess that's a new tranche of data, which I didn't realize we were about to get, but which I think are about to get. Laura, do you know all about this?
We already have it.
Okay, great.
And they're going to be tested. If I'm correct, the initial testing for those compounds is happening this week.
Right, wow.
I don't know if you ever met her, by Julie. She's going to be joining them. It's going to be helping on the screening team.
Awesome. Okay, because I was talking, so Alistair Parks presented it. I think, no, it wasn't Alistair Parks presented, sorry, it's somebody else, beg your pardon. Somebody, yeah, the LifeArc guy presented it, sorry, I can't remember his name just now. And was very happy with the idea of all this stuff going towards the project, which was awesome. The compounds that you're talking about, what are they retesting fragments that were discovered?
Yes, so they were hits, hitting multiple, I think it was Mur D and Mur E, and they were hits from SPR screening.
Right.
So it's around 23, 24 compounds. I think it's 23 compounds.
Right.
And yeah, so we will test them. Okay.
That's great. So by testing, you mean reconfirm that they bind by SPR, or are you kind of trying to soak them?
It's going to be the testing, I mean, the enzymatic assay.
Okay.
Yeah.
Fantastic.
So now the new person joining us, so it will be a good test for her to try with those tiny compounds.
That's great. Okay.
Yeah, it's just learning curve.
Okay. And of course, there's no structural information, right?
Say again?
There's no structural information.
No, not yet. But they can be incorporated once I have enough APOs. Yeah.
Okay. All right, great. Sorry, Joe, you came in.
No, just to clarify, the LifeArc compounds have been identified through SPR.
Yes.
Correct? Right. And so now you're trying to do enzymatically to see if they're functional and so on. Yeah. Okay. And you said when the structures, I mean, so LifeArc hasn't disclosed the structures. There's some paperwork that has to happen. Is that what I heard you say?
I can check that with you now.
Okay, that's fine. It wasn't quite clear when you said something about the structures, if they're actually.
Oh, I thought you meant the protein structures of it.
No, just the structures of the compounds themselves.
Let me find.
They've not been disclosed. This is a LifeArc project, and they are happy for them to be disclosed.
Yeah.
But I think that it just needs a nod from Warwick, right?
I do have the structures. I have the smiles. So they have been released to us.
Okay. I mean, I'm happy to, get written approval of all this by email to make sure everyone's on the same page, but the conversation at the Gordon conference was really positive.
Good.
Sounds like it'd be a good idea to have the paperwork in place just in case.
Yeah.
Maybe you can talk to Chris because it might be already done.
Yeah. Okay. Okay. But yes. It sounded as though this was separate to a pre-existing LifeArc project that Chris had been involved with years ago.
So this is like the recent data that they sent us. These compounds arrived a few weeks ago.
Yeah.
Yeah. So
Okay. Yeah.
I'm guessing it's the updated list.
Yeah, I think there are two things going on here, and this new thing is a fragment screening project.
These are quite small compounds from what I could gather.
Yeah.
But yeah, you can double check with Chris and see if things are up to date, and
Yeah. Okay, I can do that offline if he's not going to join us. That's fine.
I don't know, because he is doing something in Bristol. It's a conference there, and he's attending.
Got it. Okay, I'll put that on my to-do list.
Yeah.
Okay. And the other thing that came up was the accumulation guys, are the people who are measuring compounds accumulating in bacteria and who are trying to formulate rules for how to make compounds accumulate. So the Hergenrother lab and other labs that were there, they're all interested in trying to work with us to try and improve compounds that we're designing. Certainly the Paul Hergenrother himself was interested in helping out. He's got a student who really wants to participate in the project, so I'm going to reach out to them. I think some of the feeling at the conference in that group was that we should be trying to design in accumulation characteristics as early as possible, rather than trying to fix compounds that are good inhibitors and then making them accumulate. So to try and sort of prioritize molecules that will in any case accumulate, which is interesting, and certainly we can work with them to try and help do some compound design. But generally speaking, experimentally, it sounds as though there is help there if we wanted to have accumulation characteristics or accumulation, data kinetics measured through a collaborative endeavor. It sounds like there are people there who can help us. I heard that there are groups in the UK that do this too, and that might be more straightforward, but it'd be useful to have a sense of which group it might be good to work with. If, say, we had 50 compounds, for example, the enamine compounds that we've been talking about, or Yuhang's compounds that he's making, who we might want to approach to measure those if we wanted to do that earlier rather than later. Anyone got any strong feelings on measuring accumulation?
I guess if you want to improve the data set, then you need to make sure it. I was going to say, if you want to improve the data set, then you need to make sure the assay is exactly the same, no matter where you get it done.
Yeah.
So I'm quite familiar with this group. So I actually worked with when I was part of the IMI several years ago when I was at AstraZeneca. So I worked with Paola Ricciaroni, who's in Italy, and done all the computational side. But I think Helena out in Oklahoma would I think her lab must be doing the accumulation stuff. So I would think that I think she's the PI on that NIH grant, and I would say she's probably the person to work with. I've been at meetings with her and stuff. But anyway, I would say that would be the person to. But I guess in terms of I guess it's a catch-22 thing. It's like, when do you start worrying about measuring accumulation without MICs?
Yeah, exactly. Yeah.
So I think it's that's the question that to kind of try to figure out what the team wants to do around that, because you don't have MICs. These, first of all, the compounds aren't that potent at this point. I understand, this is the whole, like, when do you do these things, right? Yes, you want to try to build in the characteristics. I mean, I know, that article that was published, again, I think that came out of that big NIH grant, some of the rules. So anyway, I mean, I think that's something obviously to think about is if you're doing chemistry, because the next steps, right? I guess, if we get structures and then you get a new grant that supports more chemistry resources and you start that design process. So, but that's great to make that connection, Matt. I think that's, and that may be something you actually, as you think about new grants that you're, want to get more funding for, they may be good co-investigators to be part of those grants.
That's right. Yeah, there was just lots of sad
especially
stories
if you're doing for example, with Helena, basically, having that sorry to sound provincial here, but having U.S. collaborators if you're trying to go after NIH grants, I think is probably a good thing to have.
For sure. Yeah. Yeah. Just incidentally, some of the most interesting talks are about the porins, the people who've been doing modified bacteria lacking any porins whatsoever, and finding the compound still got in. It was really interesting.
Yeah. I mean, again, this was a senior moment here, but yeah, this is again part of the IMI. We had some of those experiments, and yeah, I mean, there's some head scratchers around, how much of it's passive diffusion versus active porins and so on. So it's good this kind of work's going on, continuing to happen, because obviously it's important.
Yeah. Okie dokie, leave that with me, and I will try and make some reach outs for accumulation stuff. In the case of Yuhang's compound, I mean, it's it would be great to get those measured anyway. So the difference of an OH to an NH2 should make a difference to accumulation. If we can have that measured, that would be awesome. But more generally, we can think about it in terms of compound design going forwards. All right, I wanted to get, so there are a few key things today which I've highlighted here. So I guess Laura, if you're happy to update us on the first thing, which was what's been going on the last month in terms of the enamine compounds that have been measured.
About the screening and all that.
Yeah. So what anything that's happened in terms of you shipped stuff to Diamond, and you've been looking at further at those compounds.
So okay, let me share. Maybe I can share my presentation. Can you see the PowerPoint?
Yeah.
Whoop, I can't. I'll do it now. It disappeared. Right. Can you see it now?
Yeah.
Okay, so yeah, I've done a few things to try to get the structures with the hits from the enamine library. But I haven't been able to obtain any ligand bound in the structures that I collected, or the data sets that I collected from Diamond. So basically at Diamond, I got crystals from Mur E, E. coli Mur D, in the presence of obviously the 4 hits from the enamine. Then E. coli Mur D with ADP melted completely, so I couldn't send any crystals. I also sent crystals with UMI without magnesium, and 2 versions with magnesium. And the compounds are, these ones, the ones that we are calling, these are the names of the compounds for now. Different concentrations of compounds, and as well different soaking times, right? But I couldn't see any ligands there. The only thing I could see of something happening was in the crystals from Mur E with JO6, and they were showing a more close conformation than usual. We have seen this before, if you remember. And the rest of the thing is no new crystals yet, but I have done different rounds of co-crystallization. The last one is pretty recent, so I'm still hopeful that something will come out from that. And now I'm running out of protein, so I need to make more protein to be able to do more follow-ups. Have also set up some more crystallization plates for doing soaking on different compounds, whatever we get and whatever we have, including the old conditions and new conditions for the E. coli Mur D with UMI, because there was a secondary condition that also gave me a structure. And I'm also looking into that one. That one, I'm still waiting for crystals to grow. They took a bit longer to grow in the first initial screen, so it's still within the time of them to be able to grow. I think that's it. And these compounds are going to also be tested on the assay against Mur F as well, to see if there is any extra inhibition. That's going to be done when Adria comes back from holidays. I'm thinking 2 weeks time. And then there were more things about essays. if you want me to continue talking about essays.
Sure.
Actually, I was just questioning, Laura,
Yeah.
I know you and I talked offline about trying to test these, I mean, using a control like ADP or something in terms of, repeating, I guess a control compound or something smaller.
Yes.
I don't know if you were able, I think you said you were trying to source the compound or something, or I was just a comment about, I guess in theory, in terms of these like soaking experiments, just confirm that, I guess, quote, a positive control to use.
Yes. So I'm making a list of them and then I will buy them and get them. The thing is that I don't have enough protein for testing it.
Yeah, I understand.
Yeah,
so I'm saying going forward.
So I have paused a little bit that until I get the protein, but yeah. And I'm also doing some other things that
Right.
Yeah.
Okay.
That's yeah.
Did you see any difference in just in terms of the compound concentrations? I mean, this whole melting and precipitation, I guess more the bigger issue is, the precipitation out. And I guess you're using like some, 2 or 3% DMSO or something like that.
Yes.
Some, but like the 50, like the 50, that's millimolar, right? So 0.05 millimolar. What is that? Yeah. That's 50 micromolar? No, that's 50.
50 micromolar was the lowest one that I tried.
Right. And I was just wondering if you saw any better behavior at that point?
No,
or
I think
it's all kind of similar.
So there was less precipitation of the compound, right? So there should be more in solution. However, if that's enough to get the crystal, the compound bound into the crystal, then that didn't work out. So, but yeah, you can see a progression on compound aggregation and compound concentration. You can see the correlation on that. Yeah, so I think it's just the condition is not ideal for this. So yeah, I'm hopeful the new condition will make it happen.
So in these, sorry, again, I'm just, so they do these diffusion experiments or something where you have like the solution of the compound and it basically through like diffusion, you understand what I'm saying? I think there's these methods where you have the crystals and then you have your compound in like a reservoir and there's like a diffusion kind of experiment. So there you basically, you're not putting the compound directly in, it's more like a diffusion kind of experiment.
So yeah, I could try that as well, but it's not very high throughput though.
Well, we're not, I mean, the question here becomes, I know we're trying, it makes sense to get like an XChem system, but, on the other side of it is like getting, we haven't been able to get any kind of structures. I mean, just getting a
but I will need to buy as well other things for that. But I did, so I did different tests on the concentration and the crystallization condition, but they were all, showing the same thing. I just trying to think if that's mimicking the dilution. So as soon as they were touching the condition, when I was looking at the microscope, as soon as they were touching the condition, it will aggregate.
So Yuhang, do you have any comments around this kind of, suggestions or this, idea about this diffusion thing? Is that something that makes sense, or that's kind of really grasping at straws?
Is he in today?
Jan's with her. Yeah, he's with us. He may have stepped away.
Oh, okay. We'll
just
come back to that later.
Just while we're waiting, I had a question about aggregation. When you say aggregation, do you mean that the compounds precipitate?
Yes.
Okay. Right, because I mean, of course, it's a feature of some of these molecules, right? Is that they may not be terribly water soluble. Is that the issue?
Well, this condition has a lot of things inside, so it could be anything.
Okay. So it's not like a criticism of the compounds that we're using that we should be.
No. It's a criticism. I'm criticizing more the crystallization condition than the compound. Okay. At this point.
Fine.
Yeah. So that's why. Yeah.
Sorry, question was asked. I had to get off mute and I'm still biking.
Oh,
no worries.
In a couple of minutes.
Yeah, don't worry. Don't bash into something.
I don't quite understand what kind of diffusion system you were referring to.
It's like
I'm
the
sorry,
capillarity.
again, I mean,
it's like the capillarity crystallization.
Okay, yeah. We have tried that a little bit. It's pretty finicky, and it can be very, very difficult to retrieve crystals out of that system. That has been our experience in the past. So it's a last resort, but I wouldn't take it as my first, second, or third choice.
Thanks, John. Thanks. Like I said, it's just, again, something I've seen in the past in the literature or whatever. So I was just wondering if it all makes sense or not. So it sounds like it's, as you said, the last resort kind of experiment.
Yeah. But it could be something like a look into. I can ask a Diamond if they have some capillaries instead of having to buy the whole thing. Yeah. Yep.
Okay, perfect. Thank you.
And yeah, in terms of, there were a few comments.
Yeah, you'd ordered some new enamine compounds. I guess they're being made.
So the compounds, the hits, I've got the enamine hits, right? Those are here already.
Yeah, there were some compounds that were ordered that were around those hits. Am I right, or am I confused? So the four that did really well.
Yet. I don't think there are any yet.
Okay, fine. I mean,
I got confused myself. I thought it were, so the new compounds that I got from Ed were from the Cato library.
Oh yeah, no, sorry. I thought that from the 4 hits that you were now experimenting with from, the enamine library that Joe and everyone had constructed, the 4 hits that worked well and the multi-targeted, that there were some molecules around those that have been ordered?
No,
not to my knowledge.
No, I think there's been some compounds reordered to have new stock of some of the hits. That's what I think both Laura and then I think Laurie had ordered a few of the others that Laura hadn't ordered, but I think they were all around just the actual hits. No near neighbors have been ordered as far as I'm aware.
Okay. And there was some discussion last time of a couple of Warwick students doing some chemistry. Is that right?
They were doing some essays.
I'm
not sure. Yes, I'm not sure they have done any chemistry. So they have been helping with some essays, but yeah, I don't know how far they went because I haven't seen the last report. They just finished. So yes, and
they've just finished. Wow.
Yes, I haven't seen them this week, so I'm guessing they're not here. Maybe they're writing up at the moment.
Okay. If they want to come along to the next meeting, please do invite them.
Yeah, we will. It was summer students, so I don't know if they will be continuing. Yeah. Then so we got the compounds from Cato, from Ed. Those will be run on the SPR and essays together with the full competition essays. Competition compounds. So we will merge them together and run them at the same time.
Okay, that's great.
Yeah. And what else? There was another comment about soaking experiments. Yeah, that I'm growing new crystals for soaking experiments. I will coordinate with Lisbeth for doing more soaking. If we do more compounds into soaking, then I will go to XChem.
Yeah, and the enzymatic.
Yeah, and then regarding the
measurement of the atomized compounds.
Yes, so I have done the atomized compounds on the SPR experiments in competition with AMP-CP. So I will update that in the in GitHub.
Right.
Sorry, I can't talk today. So I will update that as soon as I can, as well as the results on the pocket from the Atomwise and the XChem screens. In summary, it's the same pocket. So the new pocket is it's the same pocket for both libraries results. And then, so we having the essay team has also been busy trying to screen Atomwise, the Atomwise library against Mur Ligases. So they did the first screen, which looked promising in the first go, but when they run the screen against the coupled enzymes, that gave a lot of hits as well, and very similar to the initial ones. So we're having an issue with that library inhibiting the coupling enzyme assay. So they need to be rerun on the other assay that we have replaced.
Okay. I mean, if the atomized compounds are doing anything, if they're inhibiting anything, then it's a very good reason for us to go back and talk to Atomize again about help and follow up.
Exactly. So I can't wait to see those results. And on the queue, and on the queue, there will be again re-screening enamine against all the Mur Ligases in case we are missing any other compound, because now we screen only against one protein and we are using those hits and see if they have potency against the other Mur Ligases. Okay. So the idea is also re-screen the enamine library against other Mur Ligases. So that's on the queue as well. And there was an initial compound sent to us a few years ago, and those will be as well included on the screening. So basically what we want to do is in the next few months, screen all the compounds that we got for the project.
Right.
Hopefully multiple years.
Yeah. Okay, great. Thank you, Laura. Is that everything?
Everything.
Okay, great. All right, thanks. Well, just so that's a good segue, because I think that I guess we can just then call on some of our chemists. So Yuhang and Ed and Daniel just to give really quick updates on some of the chemistry, because I think they've all sent or are sending you compounds.
Yep.
So Yuhang, do you want to go first?
Yes, sure. So for the compounds I made and have been updated below, and
I can share.
Yeah, sorry. So I've managed to send the micro ED samples because they require way less than the microbiology sample. I checked last week with Laura, and she told me that for microbiology, for these 5 samples, they are going to require like 50 to 60 milligrams each. So I currently have four of them having this amount to be shipped. But the only problem is the amine product is really hard to accumulate to this. It's like each time it got like 5 milligrams or 9 milligrams, but like if you scale up, it might risk the yield, so it might still ended up in getting like less than 10 milligrams. So I'm trying to do multiple. I'm trying to start like multiple small scale reactions and try to accumulate, but definitely need more time to like purify it. Then also like the NMR is down, so it's really difficult.
So these 3. Am I right? These 3 have been sent on small scale to Warwick. Am I right?
No, not being sent.
Okay, because if this molecule doesn't inhibit any of the Mur ligases in vitro, then is there any point in trying it on that scale?
Oh, in like microbiologists.
Yeah.
You mean like we can do the assay first?
Well,
like,
yeah. I mean, is there any possible way in which this thing will be lethal to anything if it doesn't inhibit any of the Mur ligases in vitro?
I do have 5 milligrams like ready to go, which have been already examined by NMR high-res.
Yeah. I'm just thinking that the in vitro first, and if anything hits, then we do the big. Yeah. Because otherwise, it's going to take a long time to make 50 mg.
Yeah.
This is Joe. Sorry. This kind of blows my mind. The requirement for 50-60 milligrams for doing the micro, what's going on there? Because I mean, sorry, I just talk about my 15 years at AZ doing and I wasn't in the microbiology lab, but chemists were never making 15-60 mgs to do microbiology. So
I can talk to them again. This is
I mean, they should be able to do
what
this.
Adrian told me. So
they should be able to do something like 2 to 5 mg should be enough to do the microbiology. I don't understand what's the requirement here for such a large amount of material for doing some simple micro experiments against, a couple, you basically want to be doing Pseudomonas here and maybe E. coli, like two organisms and, a couple different concentrations. And I just kind of, anyway, sorry, but I just,
yeah,
I think that's an unreasonable, in my view, an unreasonable demand of amount of material for doing microbiology experiments.
So we have not been able to talk to John about this. So this is the number that Adrian gave me from other, I'm guessing from other places that they have been running. So yeah, I don't know where the number comes from. That's what I mean. Anyway, so I can't say anything about where the number comes from because I don't really know. Okay. It felt a lot to me as well, but yeah. I'm not familiarized with those experiments either. So
seems to be 10 times too much in my opinion.
Yeah, we're not doing human trials here.
Let me follow up with John. I think he is feeling better, and let me follow up with him.
Okay. All right. But in the meantime,
send us lower scale so we can run the essays.
Yeah.
Okay. Right. Then the essays should be fine. Oh, that's a lot of pressure off. Thanks.
And here also the SS you're talking about SSGCID samples, right? This is all 10 to 15 mix. Is that right?
Just a double check with Yang, like he's here today. So I just want to double check with Yang if that's the correct amount required for.
So you want them to be used as soaks for soaking experiments?
Yes.
Yeah, I think 5 mg would be plenty.
Okay.
Cool. But I don't know who to send these samples to. Bart, I saw Bart is on the phone.
Yeah. Previously we sent to Bart. Yes.
Okay.
I guess the same.
Because in 22 days, our gig will come to an end here.
Right. Is it available to receive these samples and store them in the fridge, like maybe -20, something?
I let Bart speak.
Sure.
I couldn't quite hear what you said since I don't know, my audio wasn't great, but yeah, we're available to receive compounds. We did actually get our notice that we'll be refunded Monday, I believe, so that we can continue.
Awesome!
That's amazing. Congratulations.
So much.
Yeah, that's awesome, Bart. I'm sure it's been a very stressful time. Wow, that's great.
That's so
yeah, it was pretty close. I mean, because we end in 3 weeks, so it was kind of at the last minute. But so we'll be able to continue working on it, and we should arrange the transition of receiving materials.
Awesome.
Yes, that's everything.
Okay, great. Thanks, Yuhang. Guess I just got to get these things in the mail. Right. All right. Okay, thanks. And then also, Ed, Daniel, did you want to update on the competition entries?
Yeah, you just go to that summary one.
Where is it? It's 85 or something, isn't it? Sorry, I should know where that is. Wait a second. Yeah, I'll try and bring it up. Hang on. There.
Yeah. Okay, so I think Laura already has 2 of the blue enamine compounds, but the rest of them, I just got an email today saying that they were ready, except for that top right one, which they were having trouble purifying. So we're just gonna leave that one out. In terms of the other pink ones, so I've been working on those top 2 in Jan Jensen's box. I've made the right one. I just need to do 1 more purification. The left one is a bit of a struggle. I think it's because of this ring in the middle. It's not behaving too well, but I'm trying to get that. And then in the UCL CompSci box, that's we have to make, but that one got shipped to Laura, so she has to send that to us. Then we can do that one reaction. And then in the bottom box, the middle one, I've also been having a bit of trouble with the reductive emanation, but I've found some other conditions, so I'll try that. And then the one on the right in the middle, I've made and just need to do one more purification. And so the other three in that top box are Daniel's ones. So
these two are the ones that were shipped already, sorry?
Yeah. So Laura has those already.
Yeah. So this is these three.
Yeah.
Those three I'm working. I have the core rings, the first core ring and the second one. And I'm currently working the third one, core ring. My biggest problem here is just how to synthesize the diol. I repeat the reaction a couple of times and the diol since it's not working. I found a new strategy I will try this week, but so far the core rings is ready. I just need to fix the problems with the diol synthetic pathway.
When you say the chlorine, sorry, you mean here?
Yeah, I have a bromine. Yeah, I have the bromine there. Yes, I just want to do substitution reaction.
Right.
And the second, that one I'm working now to do. I have the nitrile group there. I need to do the amine, and then keep going with the synthetic. It's three more steps.
Okay, so according to Eyermann, they're going to ship the blue ones now, right, Ed? So that means that we'll have them in a week or something, right?
Hopefully, yes.
Yeah. All right. So then, and those will be going directly to Laura.
Yeah.
Okay.
Yeah.
All right. So I guess we can do the majority of these things soon. So I guess we need to try and, if we're going to polish these off, try and polish them off in that kind of time frame, because then if Yuhang's shipping those compounds to Laura and if these are being shipped, then it would make sense to do those pretty soon, all at the same time. So I guess it gives a little bit of a time frame after which we say, well, enough's enough, and we just screen what we have.
Yeah,
I think. Laura, is there any particular constraint for you on screening all these molecules?
Yes, the SPR is heavily booked at the moment. So if I know, at least 2 weeks in advance, so I can make a full week booking.
Right. Okay. But does it then make sense to try and get that booking in about 3 or 4 weeks or something?
Yeah, I have been booking every 3 weeks or something like that.
Okay.
And then canceling the booking just in case. But
okay. Yeah. You must be popular.
I'm not being very popular on SPR at the moment.
Okay, well, so maybe we should aim for the end of the month kind of thing, then, like the last week in August or something.
Right.
Is that would that be doable?
Yeah.
Okay, because then we can try and get
I've got everything I need for the SPR.
Okay.
So.
Okay. Let's say last week in August. Let's aim for that.
Okay.
Okay, sounds good. Thanks, guys, for the update. We've got like 15 minutes left, so I wanted to make sure that our structural biology friends were able to update us on 2, I think, new structures. And I just want to go back to where we were here. So this is August. All right. So last time, Jan, you gave us an update on some of the stuff you were trying on. Wait, I'm not sharing screen anymore, am I? Hang on a second. One moment. Last time, Jan, you were telling us about some work you were doing on these 2 structures, and then separately, I think one of these is the same as a structure which came through. I got an email from Bart, an auto email from Bart, talking about this structure. And then we previously been speaking about this structure, which was being held back. Jan, do you want to summarize this much more clearly than I possibly could?
Yeah. So the first one Pseudomonas Mur D with UMA that has been deposited and it has been shared with the team. So that's all done. Then the Pseudomonas with Mur C with the compound listed in the next line. Best to my knowledge, Peter Horani has followed up on that, and that structure has been deposited. I can't give you the PDB code. Oh, there it is. Okay. Yep.
It's been held, so it's not there. And I guess we were having a discussion about whether that was the best strategy for it. I mean, I think the assumption from this group is that everything just goes straight up, unless there's a reason to hold it back. I guess I wasn't sure.
I really don't. I couldn't follow Peter's logic, because my understanding of this group is that everything gets published right away.
Yeah.
I think he wanted to be extra cautious and brought up the option.
Right.
But in my opinion, that's the message. That's the remit of this group. It's the remit of SSGCID to deposit and make it public.
Yeah.
So
do we just need to give Peter the go ahead to unlock it and deposit it?
I think so. Yeah.
Right. Fine.
I mean, it's deposited. It just needs to be released.
Right. Yeah. No. Fine. I think that was on us. We were just wondering why that was.
Yeah.
There was just a bit of confusion there, but that's great.
Yeah.
So that's Yuhang's command in there.
Yeah. Yeah. And then very recent news is, or could I get permissions to share my screen really quick?
Oh, sure. Yeah. Please go.
So I got a new structure of Mur C with this AstraZeneca compound here. That was just collected a week ago, and let me just double check. This is AZ1364498. And we got reasonably good data. We had a bit of a similar problem of what Laura described, that crystals were melting upon soaking. It took a careful titration of time and soaking time and soak concentration of the compound. And once I had that under control, we got for this one 2.2 angstrom data. Curious fact was, I thought I had found a new crystallization condition that only had one molecule per asymmetric unit. Once I tried to reproduce this crystallization condition, most of the crystals had 8, at the old crystal form with 8 copies per asymmetric unit. Nothing wrong with that, it's just a lot more tedious to work on 8 molecules instead of just only 1. But I'm in the process of refining the structure, hope to get this in peer review today or tomorrow. And then we should have that structure shared with the team very quickly. I have another set of
something.
Yeah.
Sorry, just in the middle of your screen there, there's a yellow dashed line between what looks like an NH2 and the carbon in the middle of the molecule. Is that right?
Yeah, that's asparagine. It just, the yellow dashed line indicates it's a bit close for what you would expect, and it's a non-favorable interaction, but that's what the data shows us.
Non-favorable, fine.
It's 3.16 angstrom, so it's pretty long, so I wouldn't be too concerned about that.
Right.
But very nice. We got pretty well-defined compound. It has a couple of floppy bits, so they take a little bit of time. But the crucial bit of the protein, the ligand binding site, is quite nice. And
that's Mur C. Fantastic.
That is Mur C, exactly. And then I have another structure at similar crystal or same crystal form at lower resolution with another AZ compound. I also co-crystallized, got new crystals, and they will go out to the synchrotron this week for collection on Friday. So hopefully we get some decent data then, or I can try to refine the lower resolution, which is 2.9 or so angstrom. Ideally, I'd like to get a bit higher resolution to see a bit more how the tail of the compound is oriented. So we're on the home stretch for SSGCID, but it's still things are going on.
So just a quick observation, just for those kind of molecular recognition folks. So you notice in this that there's actually a carbon to the the asparagine, the conserved asparagine. So where your pointer was, it's 2.91 angstroms from the carbon.
This one
right.
is
no.
on the right.
On the right.
Oh, this one here.
Yeah. So you see this in kinases where you have an aromatic CH bond forming kind of a pseudo hydrogen bond to a hydrogen bond acceptor. So that 2.9, normally you would think of that as being a bad van der Waals contact. But in fact, this is one of the things that have been observed when people are starting to do a lot of kinase hinge binders, where you get aromatic CHs forming, quote, these kind of hydrogen bonds to hydrogen bond acceptors. So this is an example, like in this case, in asparagine, instead of it being a backbone carbonyl, it's actually asparagine side chain carbonyl. But anyway, it's just a comment around molecular recognition and these, quote, non-traditional hydrogen bonds for between an aromatic CH and a hydrogen bond acceptor. So,
but it's interesting because we previously discussed the idea of a purine analog, right, where that would be converted into something that's favorable potentially, even though the geometry is a bit wrong, right? But that would ruin the favorable contact that's coming out northeast there, right? Because that would be turned into a CH, right? Yeah, but it goes a little bit to the discussion we had previously about purine analog that Yuhang was thinking about. The long side chain, it's pointing north up on the left-hand side of the model. That's out of dissolvent, is it?
It's into the solvent. In this case here, it's butting against a crystallographic mate.
Yeah.
So fine. Whatever is in gray color here or in light color, that's a crystallographic neighbor.
Fine. Okay. Beautiful.
Yeah.
The
Excited that
amine looks like it's forming a salt bridge.
Exactly. Yep.
Up the top.
Yeah.
Yeah. With this aspartate 229. Yeah. But I will share that structure once it's all finished. This is work in progress here.
Thanks, John.
Okay, that's all I have here.
That's wonderful. Thank you. There was a comment you made last time about Acinetobacter Murdi with UMA. I can't remember if that was a structure that we had obtained. You've got a number of crystal forms.
Yeah, but I never had UMA bound.
Without the UMA, that was it.
Yeah. They were grown in the presence of UMA, but we could never see any UMA.
Okay. All right, great. Look, I think that those are the key things. And does anybody want to chip in with anything we haven't discussed yet? Given the time.
Laura, I guess, so we have a minute here. So Laura, so I know one of the things that Warwick has been kind of trying to work through is actually the analysis of the assay data. I know Chris made some comment about trying to get some more, I guess, computational or macros, whatever it might be, to kind of be able to process the data, because I guess it's been quite tedious to analyze the output from the assays. So do you have any sense of if any progress has been made on that?
I think it's sorted, at least from the last time I talked to them.
That's fantastic.
Yeah, so the last enamine screens, it has been done with this LabMat script that they have created. So it should be, now it seems to be easy. I don't know if the data at least much easier than before.
That's great. That's great.
Yeah.
All right, thank you.
No problem.
Okay, that's great. If there's nothing else, then we can call it a day. Next meeting, wait, it's right there. It should be in the diary already, September 13th, 2 p.m. Again, please let me know if there's someone who is not being invited to these who should be invited. I can include them on the diary invite, make sure that they're there. Please don't hesitate. Jan, it would be great if you can come, but we understand that your involvement is now subject to other pressures. So if you feel you can't make it, please just say.
Yeah, I think I might still have a structure to present, so I might as well join that meeting and then.
That would be great. Awesome. If you've got the time,
yeah. Oh yeah. It's early in the day. It's 6 a.m. It doesn't compete with many other meetings.
Right. And Bart, congratulations again. That's really great news.
Indeed.
Yeah.
Yeah. All right. Good to see everybody. Thanks so much for coming.
Thank you.
Thank you, everyone.